Why is an HIV vaccine so hard to develop for immunity? Have you ever wondered why creating a vaccine for human immunodeficiency virus is such a tough challenge? Let's break down the reasons behind this complexity in a straightforward way. First off, HIV is unlike many other viruses because the immune system does not naturally clear it. When someone gets infected, their body does not develop lasting immunity. This makes it hard for scientists to figure out what kind of immune response a vaccine should trigger. One major hurdle is the extreme diversity of the virus. HIV mutates rapidly due to a faulty enzyme called reverse transcriptase. This leads to a wide variety of viral strains. The virus constantly changes its surface proteins, especially the envelope proteins that the immune system targets. Because of this, a vaccine must be able to prompt the immune system to recognize many different versions of the virus at once. Another challenge is that we do not fully understand how the immune system can control or prevent HIV infection. In other diseases, scientists can identify specific antibodies or T-cell responses that protect against the infection. However, people infected with HIV usually cannot clear the virus, and there are no clear immune markers that predict success for a vaccine. This uncertainty complicates the design of an effective vaccine. Additionally, HIV establishes latent reservoirs very early in the infection process. It integrates its genetic material into the host's immune cells, like CD for T cells, and can remain dormant. These hidden reservoirs evade detection by the immune system and current treatments. For a vaccine to work, it needs to generate a response that is faster than the virus's ability to set up these reservoirs. HIV also has clever ways to evade the immune system. It uses heavy glycan shielding on its envelope proteins, which hides important sites from antibodies. This makes it difficult for the immune system to neutralize the virus effectively. Moreover, traditional vaccine methods that use weakened or inactivated viruses are not safe for HIV. The virus can integrate into host DNA, which raises safety concerns. There is also no small animal model that accurately predicts how a vaccine will work in humans, making early testing a challenge. Current research is focusing on inducing broadly neutralizing antibodies that can target multiple strains of HIV. However, creating vaccines that reliably stimulate these antibodies has proven to be a tough task. In practical terms, a successful HIV vaccine needs to prompt strong antibody and cellular immune responses that are broad and rapid enough to prevent infection or control the virus before it can establish latency. This requires innovative designs, like Mosaic vaccines that combine different viral components, and a better understanding of how the immune system can control HIV. From the immune system's perspective, HIV is a tricky target. It constantly evolves, hides in host cells, and avoids the usual defenses that vaccines aim to activate. This makes the quest for an effective vaccine a complex challenge that continues to demand extensive research and new strategies.